Hello students and my dear friends, myself Madhu Vancha. In this session, we'll discuss some of the important MCQs which will be helpful in the lab technician recruitment examination. First question, identify biochemical reactions out of the following pairs which makes cholera red reaction. Option A, indole and nitrate reduction. Option B, methyl red and citrate utilization test. Option C, Voges-Proskauer and urease test. Option D, glucose and mannitol fermentation. So here, which pair of the reaction makes the cholera red reaction in Vibrio cholera? So here the right answer is option A, indole and nitrate reduction. So what is cholera red reaction? Cholera red reaction, this test is performed by adding pure sulfuric acid dropped to a 24 hours peptone water which is having the culture of the bacteria. So in this test we add pure sulfuric acid dropped to a 24 hours peptone water which is having the culture of the bacteria. So in case of positive reaction, red color reaction appears immediately. If in case of positive, red color appears immediately. So this reaction depends upon the production of indole and nitrates, and the red color develops due to the forming of nitroso-indole. So here the right answer is option A, indole and nitrate reduction test. It makes the cholera red reaction. Next question, the urine preservative which causes false positive reaction in protein. So which urine preservative causes false positive reaction in protein? Option A, formalin. Option B, toluene. Option C, boric acid. Option D, thymol. Here the right answer is option D, thymol. It causes false positive reaction for albumin, that is protein. Next question, fruity odor in urine is due to the, so fruitish odor, fruitish smell in the urine is due to option A, glucosuria. Option B, bacterial infection. Option C, chaluria. Option C, ketosis. Here the right answer is option D, ketosis or diabetic ketoacidosis. Fruity odor, fruit-like smell, we can see in case of diabetic ketoacidosis. It is the dangerous complication of diabetes. So here when this one takes place, it takes place when the ketones build up in the blood. And not only in this condition, so other conditions such as maple syrup urine disease. So in case of maple syrup urine disease also, we can see the like fruity odor. So what is maple syrup urine disease? It is the rare condition and life-threatening genetic condition like which prevent the body from breaking down some of the amino acids. So it mainly affect the babies and children. So here in this one, the right answer is ketosis. Next question, the byproduct of incomplete fat metabolism is known as option A, microalbumin, option B, urobilinogen, option C, lipid, and option D, ketone bodies. Here the right answer is ketone bodies. So the byproduct of incomplete fat metabolism is like ketone bodies. Next question, peptone is in fact, so what is mean by peptone? Option A, agar-agar. Option B, proteases and amino acids. Option C, polypeptides. And option D, both B and C, which means that is proteases and amino acids and polypeptides. So here the right answer is option D, that is both B and C. So what is peptone? Peptone consists of protease, amino acids and polypeptide. So peptone, it is a complex mixture of partially digested proteins. So how this peptone is obtained? That is obtained from the lean meat or heart muscle, and it is digested by proteolytic enzymes. It contains proteases, amino acids, polypeptides, phosphatase, minerals, and growth factors. So what are the growth factors? Nicotinic acid and riboflavin. Next question, crystal violet blood agar is a selective medium for option A, streptococci, option B, staphylococcus aureus, option C, Corynebacterium diphtheriae, option D, Mycobacterium tuberculosis. Here the right answer is option A, streptococci. So crystal violet blood agar, it is a selective medium for streptococci. So it allows the growth of only streptococci and it suppresses the growth of other bacteria. So generally streptococci is resistant to the crystal violet. So to grow the only streptococci while preparing the a blood agar media, just we add like a crystal violet, so that this crystal violet will suppress the growth of other bacteria and it promote the growth of streptococci. Next question, thioglycolate broth is used for culturing. Option A, fungi. Option B, Haemophilus. Option C, Streptococci. Option D, for culture of anaerobes. Here the right answer is for culture of anaerobes. So if you see thioglycolate broth, it consists of sodium thioglycolate. So this thioglycolate broth, it helps for culture of anaerobes. So how it supports the growth of anaerobes? Because this is sodium thioglycolate, it consumes the oxygen. So as it consumes the oxygen, it allows the growth of obligate anaerobes, strict anaerobes. Next question, disadvantage of fluid media includes. So what are the disadvantages of liquid culture media? Option A, unsuitable for isolation of organisms in pure culture. Option B, motility of bacteria can be studied using hanging drop preparation. Option C, bacteria grow very well in 3 to 4 hours. And option D, all of the above. So here the right answer is option A. So it is unsuitable for isolation of organisms in the pure culture. So if you use the liquid media, so liquid media is unsuitable for isolation of organisms in the pure culture. So what is meant by pure culture? Culture consists of single type of microorganism is called as pure culture. So by using of liquid media, we can't isolate the pure culture cell. So these remaining two are advantages. So for checking the motility, by hanging drop preparation, we need liquid culture media. And in the liquid culture media, the bacteria grow very rapidly. So B and C are advantages and option A is disadvantage. Next question, the MIC value is, what is mean by MIC? Option A, the highest concentration that inhibits the growth. Option B, the highest concentration that allows the growth. Option C, the lowest concentration that kills the growth. Option D, the lowest concentration that inhibits the growth. So what is meant by MIC? Minimum inhibitory concentration. MIC means minimum inhibitory concentration. So here what is the answer? The MIC means that is the lowest concentration of the drug which inhibits the growth of the organism. So if you see option C, that is also the lowest concentration, but that kills the growth. So option C is MBC, that is minimum bactericidal concentration. So MIC is minimum inhibitory concentration. Next question, to select cholera causing bacterium, Vibrio cholera, the pH of the medium is maintained at a pH of option A 8.5, option B 6.7, option C 8.9, option D 3.5. Here the right answer is option A 8.5. So the pH for Vibrio cholera, for growing of the Vibrio cholera, the pH can be maintained at 8.5. So it can grow even in the alkaline environment also. Next question, Fletcher's selective medium is used for culturing. So what is the purpose of Fletcher's selective medium? Option A, Mycobacterium tuberculosis. Option B, Leptospira. Option C, diphtheria. And option D, Shigella. Here the right answer is option B, Leptospira. So Fletcher's Leptospira medium. So that is the selective medium for like a Leptospira species. So this medium is added with the serum and that is used for isolation, cultivation and maintenance of Leptospira species. So what is the composition of Fletcher's selective medium? It consists of peptic digest of animal tissue, beef extract, sodium chloride and agar, and the pH of culture medium should be maintained of 7.9. Next question. Mhs medium is an example of option A, selective medium, option B, transport media, option C, differential media, option D, basic media. Here the right answer is option B, transport media. So what is meant by MS medium? It is an improved transport medium. It consists of charcoal and it prolong the viability of pathogenic organisms. Actually, what is the purpose of transport medium? Transport medium, it do not allow the bacteria to multiply, but it maintains the viability. So MS medium, like it prolong the viability of pathogenic organisms. And if you see, it is a semi-solid medium and it is a modified Stokes transport medium. Next question, which of the following microorganism is eliminated in canned foods? Option A, Mycobacterium tuberculosis. Option B, Lactobacillus. Option C, Coxiella boneti. Option D, Clostridium botulinum. Here the right answer is option D, Clostridium botulinum. Clostridium botulinum, it causes botulism. So it produces like a very potent lethal toxin. It is the most powerful toxin. So here in case of canned foods, so this Clostridium botulinum is eliminated because canned food, it is a source of this infection. So it is to like it is very important to eliminate this bacterium from the like canned foods. Next question, in the gram staining process, what is the role of iodine? So iodine is the moderate stain. What is the purpose of iodine? Option A, to form a complex with crystal violet. Option B, to counter stain the cells. And option C, to decolorize the cells. And option D, to stain the cells. Here, the right answer is option A, to form a complex with the crystal violet. So iodine is the moderate stain. So it combined with the like a crystal violet and it forms a complex. Next question, the microorganisms that grow best in a low oxygen environment is called as, which means the bacteria require minute amount of oxygen. Option A, aerobe. Option B, anaerobe. Option C, A and B, that is aerobe and anaerobe. Option D, microaerophilic. Here the right answer is option D, microaerophilic. So microaerophilic, they require the minute amount of oxygen or low amount of oxygen. Next question, which of the following describes the effect of ionizing radiation on microbes? So how the ionizing radiation affect the microbes? Option A, it changes the three-dimensional structure of proteins to a two-dimensional form. Option B, it induces electrons and protons to jump out of the molecules of the microbe. Option C, substantially decreases the enzymatic activity and slowing microbial reproduction. Option D, none of the above. Here the right answer is option B. Here ionizing radiation, it induces electrons and protons to jump out of the molecules of the microbe. Next question, true about formaldehyde. Option A, bactericidal. Option B, sporicidal. Option C, lethal effect on viruses. Option D, all of the above. Here the right answer is option D, all of the above. So if you see formaldehyde, it is one of the high-level disinfectant. It is a bactericidal. It kills the bacteria. sporicidal, it kills the spores, and it show effect even on the viruses. So all these options are correct only. Next question, the organisms that can be acid fast stained is option A, Nocardia, option B, Leprabacilli, option C, tubercle bacilli, and option D, all of the above. Here, the right answer is option D, all of the above. So here, all these organisms such as Mycobacterium leprae, Mycobacterium tuberculosis, and Nocardia species, all of these are stained by the Cheel-Nielsen staining or acid fast staining. So they are called as acid fast bacteria. Next question. What is the order of reagents used in the gram staining? So what is the correct sequence of the gram staining? Option A, crystal violet, iodine, safranin, decolorizer. Option B, crystal violet, iodine, decolorizer, safranin. Option C, safranin, crystal violet, decolorizer, iodine. Option D, crystal violet, safranin, iodine, and decolorizer. So here, what is the correct order? Here, that is option B, crystal violet, iodine, decolorizer, and safranin. So if you see crystal, first of all, to the smear we add crystal violet, that is the primary stain. And after we'll discard the crystal violet, then add grams iodine, that is a moderate stain, and then discard it, and then decolorize with the acetone and wash under slow running tap water, and then add safranin. So safranin is the counter stain. So here the correct option is option B. So what is the correct order? Crystal violet, iodine, decolorizer, safranin. Next question. Why do we use stains? What is the purpose of stains? Option A, to visualize bacteria. Option B, to grow the bacteria. Option C, for studying properties. Option D, none of the above. Here, the right answer is to visualize the bacteria. So what is the main purpose of staining? To visualize the bacteria, to identify, to observe the bacteria. Next question, staining of the cell wall of bacteria by Ringrel method. What is the color of cell wall in the Ringrel method? Option A, cell wall that appears reddish brown. Option B, cell wall is colorless. Option C, cell wall appears pale green. Option D, bluish black. Here the right answer is option A. So cell wall appears reddish brown in the color. Next question, myxoedema occurs due to. So what is meant by myxoedema? Option A, hypothyroidism in the children. Option B, hypothyroidism in adults. Option C, hyperthyroidism in children. Option D, hyperthyroidism in adults. So what is myxoedema? So it is a severely advanced hypothyroidism in adults. So it is a hypothyroidism in adults. Next question, urine is formed by the following process except. So what is a formation of the what are the steps in the a urine formation, or how the urine is formed? So option A, ultrafiltration. Option B, tubular reabsorption. Option C, tubular secretion. And option D, micturition. So here the right answer is option D, micturition. So if you see what is meant by micturition, that is the action of urinating. That is the process of urination. So if you see urine formation, urine formation is done by ultrafiltration, tubular reabsorption, and tubular secretion. Next question, ozone test is used to determine option A, amylase, option B, lipase, option C, lipid, and option D, lactose. So what is the purpose of ozone test? So ozone test is used to detect the reducing sugars. So here what is the option out of this one? What is the sugar? Here the option D is lactose. So ozone test is used to determine the lactose. Next question, glycosuria without hyperglycemia is due to. So what is meant by hyperglycemia? So that is like a higher level of glucose in the blood than the like a normal level. So glycosuria without hyperglycemia is due to option A, renal tubular dysfunction, option B, pregnancy, option C, A and B, option D, none of the above. So here the right answer is option C, A and B. So in case of renal tubular dysfunction and in case of pregnancy, so we can see the hyperglycemia like glycosuria without hyperglycemia. Thank you.